First of all, thank you to the organizer for this last-minute opportunity to give a talk here. So the prelude to this talk actually starts in my PhD, where I looked at how things sink in fluid, in particular particles that are oriented. I did my PhD with Narayanan Menon, some experiments with Narayanan Menon, some theory with Rama Govindarajan and Sriram Ramaswamy. So, yeah, this work was done back in India, where I showed that if you have two oriented objects, the orientation degree of freedom can be mapped to momentum coordinate, and you can arrive at an effective Hamiltonian in the system such that when the system sinks, the orientation and position evolve such that there is a conserved quantity that remains constant. That can also, and also, when you drop two coins in fluid, you get either bound orbits like these or scattering trajectories, and those dynamics map precisely to motion of planets or comets around the sun. And for settling coins, what I showed is you can have waves of compression, dilatation, and orientation. And again, we showed that there is a conserved quantity. You can define an effective Hamiltonian, similar to what we did here. And so this is what I did during my PhD. Towards the end of PhD, Rama gave a talk at Simon Center at NCBS, and she introduced me to the biological pump. And so I applied to Manu Prakash for my postdoc through this HFSP program. And I did not know that at that time that I would be working so directly with the system, and thanks to Manu for that. 3 months into my PhD, so acknowledgement slide as well. So this work that I'll be talking about is done with Manu, Ellie Flaum, who was a grad student at that point in the lab, and Benjamin Van Mooy and Kay Beidel, who are like very hardcore biological oceanographers. And of course, there is this team of people that go in the ocean and look at various processes that are interesting to for the carbon sequestration in the ocean, which I'll talk about. And of course, I've had the opportunity to interact with a bunch of really brilliant students from all over the world that come to Prakash Lab doing all sorts of weird things. Manu calls them as a bunch of misfits. Okay, so the first thing I learned about the ocean, is that, so if you look at oceans from the satellite, you'll see this green thing floating around, and if you zoom in, so this is the photo I took in Monterey Bay, scoop out some water from the ocean and look at the richness of the ocean just becomes very direct. What you're seeing here individually are the chains of cells. There are things swimming around. There's a diverse range of shapes and also species that are present in the ocean. So the notion that oceans are just passive fluid mechanical entities is wrong. There is a rich biology to the ocean that we do not understand very well. And 50% of the oxygen that we are breathing right now, roughly, is coming from those tiny critters that are suspended in the ocean. So, and there is this like, okay, Nelson would be interested in, like, there are cone-shaped particles, for example, in this diversity. There are all sorts of shapes, diverse shapes that you can find in the oceans. And not only that they are statically rich, but they are moving around. And so it's a soup of a very complex type of active system. And although this is like not all of the ocean is like this, I should emphasize. This is a local region that we are trying to highlight, the ocean regions. So I'll talk about what biological pump is, but one thing is clear, that oceans are living at some scale. And we want to understand at what scale does activity actually become important in determining the large-scale property of the system. And I'll tell you what I mean by large-scale property as well, very empirically. Okay, so look at this planet. This is a simulation, MIT Darwin project. Each thing that you're seeing here is actually a kind of algae or plankton. So these are cells suspended in the ocean. So they grow, they sink, they come up, they move around, coupled with the fluid flows and the chemistry of the ocean. Now, satellites can give a very somewhat clean measure of how much algae are there at the surface of the ocean, but it is opaque to the third dimension, which is the bulk of the ocean. So the cartoon that people have built over the years is that you have the suspended cells in the upper layer. They have some complex interaction, but eventually they form these aggregates that are now heavier than the freely suspended cell, and they sink down to the bottom of the ocean. What that effectively does is take in carbon dioxide that is dissolved at the upper mixed layer of the ocean to the bottom of the ocean. So it sequesters carbon, naturally sequesters carbon. Yeah.
Excuse me, what is the color, the meaning of the color in
It's the intensity of the plankton.
Of plankton.
And different colors are different type of plankton. Okay, so or you can think of them as also chlorophyll maximum. The indirect measure of how we see things from the satellite is look at the chlorophyll signal. So that's the indirect measure of how. Okay, so historically, people had looked at these aggregates, like William Beebe, that literally went down in this like steel sphere, which I just see at Hui, they still have kept it. And so at that time, there were no like video, so video recording could not be done under the sea. So what we would do is have a telephone, so the wire will go up as it's tethered to the ship, and he would describe what he's observing. So his fascination was to study deep sea organisms. And so in one of his observations, what he found is, which Rachel Carson described very beautifully and poetically, that when I think of floor of the deep sea, I always see the steady unremitting downward drift of material from above, flakes upon flakes, layer upon layer, the most stupendous snowfall that the earth has ever seen. This has nothing to do with snow, as I already said, but in all the National Geo videos, you must have seen that these tiny specks that are suspended in the water, and which is what we call marine snow. They have nothing to do with ice, again. So one of the things that we can look at as physicists, which is important, is how these aggregates sink. And you might ask, okay, let's just look at these pictures and look at these dots, measure their effective size, and then use Stokes' law to estimate their velocities. So independently estimating velocity is difficult due to because these are microscale aggregates, and doing microscopy on moving things in the ocean is not a trivial thing, also because of the pressure. And these things happen like in the bulk of the ocean at 100 meters, 200 meters easily. So pressure is the issue that is relevant to imaging in these, in situ imaging of these particles. Okay, so there was a bunch of observation with cameras in the ocean. And if you try to map this simple linear relation to these particles, to my eyes, it was messy, but that's the best theorists could do it. For the last 2 decades, that people have been trying to fit a curve that relates size of the particle to the sinking speed in different ways. And I wanted to see if I can improve that understanding to begin with. All right, so there are multiple scales. So if you hang buckets in the ocean at different depths, this is the emergent large-scale behavior of the carbon sequestration that I was talking about previously. So if you hang buckets in the ocean at different depths and measure how much aggregates is collected, so instead of looking at single particles, just take an integral, what you find is that amount of carbon becomes smaller and smaller as a function of depth. That was completely empirical. People try, so the state of the art in the climate model is to fit this power law relation to estimate. And so there are several caveats there, because this power law should also actually depend on where in the ocean you're looking. But do you integrate everything? So this is the state of, this is the best you could do at this point. So, and I mean, during PhD, I also looked at like how we're looking at collective particle suspension. Historically, it has been looked at before, and I was in a fluidized bed geometry, and I was amused to know that people have done similar things with aggregates. So,
Hi, Rahul. Sorry if I missed this. Can you explain again how we know that marine snow is the dominant mechanism? Because I guess, what about other hidden unknown mechanism? Or in experiments, I can just get rid of marine snow and see how it affects the system, but obviously we cannot do it here.
You are correct. In some places, some other mechanisms could be important. For example, subduction pump, where there are upwellings and downwellings. But in the bulk of the ocean, where there are no upwellings or downwelling, other than these big gyres, what else do you have, right, to sequester carbon from the upper?
I don't know much about ocean.
But yeah, I mean, there are many hidden, so if it's about hidden parameters, there are many hidden parameters. And this talk is actually about one of those hidden parameters that I'll talk about. So let's just confine our our window to just looking at the dynamics of this particle. We are not being very ambitious to begin with, just observe, this is an observation-driven study. So this was done at steady lab setting with aggregates that were artificially created. So Manu Prakash and other grad students in the lab, while I was doing PhD, built this very clever tool to actually create a fluidized bed geometry with a spin in that you generate the upward flow by moving the boundary upwards. In a feedback control way. And it not only compensates for the downward drift, but it can also move sideways and in the subspace perpendicular to gravity. Now, this allows us to look at things that are moving in 3D while being in the frame of that moving objects. So that was a very cool invention. And when I joined the lab, I was given the task to take this instrument on the ship and look at marine snow in this open-ended manner. Okay, so we went through this is Cape Cod, the armpit, through the armpit, and then we did a Lagrangian kind of journey there, where we were just floating with the ambient seawater. And the satellite image told us that there was this rich algal bloom that was happening at that time. So we should expect marine snow. So that was nice. So there is some periodicity in this ecosystem in that every summer there is algal bloom that happens. So it's a very hot spot for studying these systems. We went on RV Endeavour, and I did not know that smaller the ship, more seasick you feel. At that point, I was very confident that, how difficult can studying sedimentation be, since I've looked at it for 5 years. But I couldn't be more wrong. So now, yeah, all bets are off in that you're, with this instrument, trying to look at microscale flow while being on this rocking and rolling frame of reference. And you might assume things would work a certain way when you are linearly extrapolating how the microscope works, but there are many issues that arises just because of this rocking and rolling. So one simple thing we could do is put it on a mechanical 2-axis gimbal, which acts as a high-pass filter, mechanical high-pass filter. So that's one way to reduce the rocking and rolling noise that comes from the rocking and rolling. This is how it works. Okay, so you place this. And the way we take these instruments in the sea now, we have done it several times. This is modular, so we actually take out each connection or wire and then take it on the ship and just set it up again on the ship. Now it's much easier. At that time, yeah, you can see that's bare wire hanging around. Thanks to Hong Chuan again for all the technical expertise that he offered to this work. So experimental setup looks like this, so which I already said. This is gimbal. Now we need a sample. So we use this giant nets, hang it in the ocean so the results I'll be presenting is 80 meters depth, and hang it there overnight. So it has collected enough debris. Now you bring it up. This is the movie of them bringing it up. And when you look at these things, this is just a gunk. Now, how do you do physics on this system? Where do you even start going about doing anything meaningful? Being a sedimentation physicist, physicists can do sedimentation. They have been doing it since the times of Sir G.G. Stokes. So just resuspended these particles. Look how these debris sinks. So you're looking at the wheel, annular region of the wheel, which is, if you look closely, now rotating upwards to compensate for the downwards drift. Can you see that? And you're also doing 3D. So this movie I'm showing just to show that the 3D structural complexity of the system. Okay, and this is highly dynamic. When you put these gunk in the fluid, there are cells, there are ciliates, there's activity to the system too, that it's not a static, fluid structure interaction. So there are flow-mediated fractions. Because of differential settling, you can get merger events. Things are coming and eating it, microbial activity, all those things are happening. Again, reduce the system to looking at steady state sedimentation of these aggregates without hydrodynamic interaction with other aggregates. So, and also it's important to know these aggregates are very fragile. You have to be very gently, take it out, put it in the suspension, and then look at the sedimentation dynamics. Now we can measure the structure in great detail through microscopy and look at the sedimentation velocity through the 2D, coupled tracking that we can do in the microscope. So it gives us velocity and shape independently. Okay, and if you try to map the structure to the sedimentation speed, there is no clear correlation in that. For the same size, you get an order of magnitude different velocity. So, which was known previously, I mean, the linear curve that you saw. But the thing is, one thing to confirm here was our data is in the same ballpark as what people had seen before in the other in situ photography and tracking, which gives us confidence that we are not doing something really dramatic to these aggregates when we are handling them. Okay, now let's look at it more closely. And yeah, what is happening? Why is there a huge variation? So if you look at flows, in fluid structure interactions, like without looking at flow, you can't say anything about it. So just pick one particle. And this is the aha moment in the work. And so what I do here is suspend tiny beads, passive beads, just to track the flow of the particle. This is a marine snow aggregate. Do you see anything unusual about this? And this is Stokesian. So there is a thing that trails behind the particle, where the beads are actually stuck. Things are not moving. And I'll play this again. The beautiful thing to appreciate here is that you're measuring microscale flow while the whole ship is rocking and rolling. You're able to do that. Also because of 2-axis gimbal, but not only because of the 2-axis gimbal, but also all the post-processing that goes in the image analysis. Yes. Yeah, that. Okay. If we, now I'm color coding the vertical velocity of the flow, you can mark this region. Okay, I started pointing there. Where you mark, you can measure the amount of this invisible thing there is. So, so it does two. So now I only showed for one of the aggregate, but this we found to be ubiquitously present to a greater or smaller extent in all the marine snow aggregates. And that's where I felt that there's something universal here in this mechanism. It's not just that we are, there's something that we can extend beyond just local, small scale sampling that could be present in, well, that also comes later when we actually find the origin of this mucus. All right. So if you measure the extent of mucus, that you can do by measuring that size of the halo, and the invisible, so I'm calling invisible size as the long axis of that ellipse that I'm fitting, and the visible size as the equivalent spherical radius of the visible part. And for the same amount of visible size, there is a range of invisible size. What invisible size is doing to the fluid structure interaction? One, it is changing the dense or buoyancy of the aggregate, effective buoyancy of the aggregate. In addition to that, it is changing the drag because of the shape itself. And both of that, so this we thought initially can explain the variance in the velocity that you observe in the ocean. And what we find is the more the mucus, the slower the particles sink. So effectively, mucus is dragging down these particles, which we observed empirically. And effective size is dramatically different. What people had been observing and estimating the sinking speed is wrong, clearly, because the effective size is completely different. And Ellie did some very specific staining to find that this mucus is actually released by the plankton. It has a biological origin. So this ecosystem that is sinking has this knob where cells release this biological mucus, which helps them aggregate, but at the same time, these aggregates become less buoyant because of the presence of mucus. And so there is a biological knob in the problem. To build a very simple, I call it a spheroidal cow model of marine snow. If you consider marine snow, so writing down the simplest possible modification of Stokes' law, where you have a heavy aggregate with a halo of mucus, where I'm assuming one-way hydrodynamic coupling, where the aggregate is generating flows that is sharing the mucus behind it, elongating the mucus, changing its shape, and the simplest, and there are some non-dimensional parameters. So remember that D is the ratio of elasticity to gravitational stresses. Density is defined as particle density minus mucus density, seawater density minus mucus density. So there's a non-dimensional parameter there. And there is another, like volume of the particle divided by the total volume, including the mucus. And this is the sedimentation time scale, and the size is essentially the size you would have without gravity. The size of the whole thing you will have without gravity would be spherical because of the elasticity. Now, in the linear response, we write down how this thing distorts. So there are two components to it. One, there is a viscoelastic relaxation coming from the elasticity of this elastic spring here. Second is how the particle shares the flow. So there is a response coming from the shear of the particle. This is to leading order in L over A. We can take only the first term here, where L is the radius of the sphere and A is the effective sphere, the sphere that is created without gravity. Because if you switch off gravity, this blob will become a sphere, and that gives an effective size. And that couples to the sedimentation dynamics. And you can find, if you assume that this is a spheroid, you can find that scaling relation exactly. Okay. So from there, what we can do, so we can measure various terms here. We can, so there are two variables and two unknowns. So through these equations, you can actually measure what is the elasticity in the system. So now the marine snow is not just a structural thing, it has material property, which is also important to this flux. Sedimentation is what we find. Okay, so from there we can measure the density of the particle, and we can, or a particular thing, and then we can measure the effective elastic modulus of the marine snow using the frameworks of those equations. And then we compare it with the experimental observations where we can measure, so there is a tail length of this mucus tail, and there is a sedimentation velocity. Based on we are not looking at marine snow in the plane, in just a size sinking plane. What we are introducing is there is a density size parameter. Everything is lumped into this parameter. And there is an elasticity, elastic modulus. And then you have the color plot. Okay. So what does it do to the residence time? So if marine snow spends more time in the upper layer of the ocean, it gets eaten away by tiny shrimps or zooplankton, and microbial degradation. And if you calculate how long these particles spend, it's 2 orders of magnitude. So it's twice the amount of time it would spend without the mucus. And also, if you plot the size and sinking, with this new model, we could show that there is some trend which is better than the original size sinking relationship, which incorporates the hidden internal degrees of freedom in the system. And also there is a rich complexity. If you look at local density of this particle, that is also very notoriously difficult to even make sense of. And that leads to some more stochasticity in the system in the density of the particle. So, for example, individual things like frustules that you see here are actually coming from the debris of the cells, which makes these silica frustules around them. And there are many other things that constitutes marine snow. And this is like a 3D tomography of snow. So, okay, the generality of the systems, is it just this one measure? So we made these observations in Gulf of Maine. Is that the only place where we find this? But that's no. We did more expeditions in other places. And we found like there is this presence of mucus comet tails in other parts of the world too. And my understanding is this is as ubiquitous as the TEP itself, which is a transparent exopolymer. So, and transparent exopolymers. So these are the polymers that the cells, living things secrete. How they secrete it is an open problem. One thing is environmental stress could induce TEP. So that's very possible. There are many other parameters that affects the size of TEP, but this allows us to do in some capacity, soft matter physics in this very messy system, I would say. Okay, so the broad picture is, so after this paper got published, Kale and Greedy wrote a perspective article, and they did some back of the envelope calculation how the Martin curve that I introduced initially, the power law curve, changes with and without mucus. So it increases, so there's a 40% increase in the atmospheric CO2 based on these observations. So if you incorporate for mucus, it is increasing roughly. Of course, these models are still at a primitive stage. We can't say much more predictively about how the CO2 in the atmosphere evolves in time, or even the measures of it. But this is the direct implications of these observations. All right, and with Arjun, I'm doing some now more cleaner, like incorporating all the degrees of freedom of the mucus, doing a DNS on these particles, including the viscoelasticity of the polymers and such. So conclusions. So we presented the direct measurement of microscale sedimentation and the fluid dynamics of marine snow in a field setting, which is infinitely more difficult than doing it in the lab. Marine snow is a composite particle, well, we all know that, but now we can divide it into like, there is a visible part to it and there is an invisible component to it, which we can directly measure. And that, its material property affects the global carbon sequestration. And the theoretical, some frameworks that incorporates its hidden degree of freedom and modifies the Stokes law that people have been using for the last 2 decades. And more broadly, my big ambition is to go towards a more physics-based understanding of this biological pump. And this is just one of the measurements that we made. There's so much richness to the ocean. And honestly, this is the first time I went in the sea. I observed something, I found something new. And it's not because any special skills that I have, it's because the oceans are rich. And there's rich physics or soft metaphysics in particular for everyone to look at in the oceanic context. And I hope that we, as a field, we slightly move towards that direction because of its consequential nature of the style of research. Thank you. Okay, I won't do that.
Thanks a lot. Wonderful presentation. We do have time, lots of time for questions. Please start.
Thank you for the beautiful talk, Rahul. Usually when I see this kind of marine snow, I often see some ideas about self-generated flow as like this bacteria or metabolically active and they generate flow. So it was great to see you can actually also see a style of flow instead of flow. But also that made me question about the system, whether it is actually metabolically active, or should I just think of this as maybe some dead or inactive cells that who already starving and just ate out of nutrient?
I think there is a separation of time scale.
Yeah.
If you look at long time scales, so there's a one-way coupling between bacteria in that the fluid flow that the marine snow generate affects the encounter of bacteria around it. But it's like, if you look at it at small time scale, just look at marine snow for 5-10 minutes, which is what I did. You don't see bacterial decomposition playing a key role. But now we have actually, in this expedition, we collected data looking at the bacteria in the ocean. So we have like how bacteria interacts with marine snow in a very clean setting, which is what I'm working on right now.
I see. And maybe one related question. Also, since these bacteria need to actually actively find food, as otherwise they will starve, there was a big literature in the ecology about how these bacteria are determined to not only aggregate, but also determined to disperse when they are out of nutrient. And seems like the mucus, at least naively, will, let's say, prevent this process. So
that also affects, so that is not even incorporated in the back of the envelope estimation that researchers have done on how this model will impact climate change. Actually, in the perspective article, they write that this will also affect how bacteria encounter and how effectively they decompose these marine snow aggregate. So that work is like, these are open direction. I mean,
thank you.
Yeah, in the first plot that you showed, when you showed the old data, it seemed to me like the different data sets kind of like organized in different lines. And I was wondering if that had to do with like the mucus properties of the different, samples that were taken.
This did not see mucus.
No, I understand. But if you would correct each one of them with some, viscoelastic properties of the mucus, will they all suddenly collapse the old data too?
So I can't say this generally for, so again, particles can form in the ocean for many reasons. The kind of particles that I'm looking at is biotic particles, primarily produced due to algal blooms. So they have some special properties. So for example, if you look at the coastline, there are lots of like pollutants that we release in the ocean. The properties of it will be completely different. So this is a special kind of particle, but at the same time, it is most of the marine snow. So the coastal carbon sequestration is just a fraction of the overall carbon sequestration that happens in the ocean. So that's the goal, to be able to correct previous observations, incorporating this. But that's, in my view, it's the lack of data. Like I don't think you can correct them without measuring the invisible degrees of freedom, which does not exist at this point. So this should prompt us to observe these systems in this micro hydrodynamic type of perspective.
And very quickly, just could you re-explain what the difference when the carbon capture, how the carbon capture will depend on the rate of falling or something like that? Is that?
Yeah. So if you, the way this biological pump is sustained is by maintaining the gradients of partial pressure difference at the atmospheric carbon dioxide and the upper layer of the ocean, which leads to the flux of carbon from the air-sea interface to the ocean, which now becomes dissolved CO2. That dissolved CO2 now gravitationally sinks, depleting the CO2 in the upper layer of the ocean, bringing in more CO2 from the top. Bacteria, what other zooplanktons or other ecosystems actually eat this away, because this is the primary production of the ocean, and all the marine ecosystem actually relies on this phenomenon. They eat these aggregates, release the CO2 back in the atmosphere. So only a part of it leaks to the bottom of the ocean. And that sets a major uncertainty, and we don't know much about those micro-scale processes in the ocean.
The faster it falls, the least it will get eaten.
Yes, yeah.
Okay, I wanted to ask, what is the biological incentive for producing this type of mucus? But maybe you kind of answered it right now, or?
So for bacteria, you can think like whether bacteria have evolved to, because bacteria also release mucus, for example. If you want to stay suspended for long,
okay,
and then you can define a cost function to it in terms of evolution, it helps you stay for longer in the upper layer of the ocean. Yeah. That can help you probably get more sunlight for these plankton, but helps them come together, agglomerate, because mucus helps them stick together as well. But yeah, there's so much that we do not know, and we need to first observe these mechanisms in this rich details.
Yeah. Thank you.
Very nice talk. So since you mentioned your previous work on this Kepler orbits, etc., have you seen anything interesting like rocking orbits of these aggregates, or some kind of multi-body dynamics, or something interesting?
I have some simulations for how, so those dynamics will be important. The nonlinear versions of those dynamics will be in that. If you look at suspension at long time scales with the stickiness in these particles, they will form these aggregates, which is what marine snow is. So How marine snow forms will depend on those details, but honestly, I did those study on coins just as a purely academic pursuit without relating it to biological pump as such. Thanks.
Yeah, I guess it's related, but so like, I'm guessing when it's...
Can I add one thing?
Yeah.
So disc-like particles are ubiquitous in that so you have literally discs-like phytoplankton that form marine snow. They're called cosconodiscus. So could be relevant, but could be not. Sorry.
Yeah, so what's exactly changing in the plankton for it to start falling? Like, is it related to the mucus or?
Yeah, so we have another paper on, so these cells can regulate their buoyancy. So in one of the expeditions in the Pacific Ocean, so we took these particles out, put in the gravity machine. What we found, I think Manu showed one of those videos in his talk, that these things can decide when to sink and when to stop, and that happens very abruptly. So there is a feedback control within the cell itself. So there is a behavior of the cells as well that plays a role in whether things sink or do not sink. But one thing is true, if cells are not neutrally buoyant in that they have a negative positive, so downwards buoyancy, if they are sticking together, forming larger aggregates, they will fall faster. Single cells might be actually, back of the envelope calculation tells you that it takes a year for things to go from the top of, given the buoyancy of the cell, from the top of the ocean to the bottom. So you need aggregation in order for benthic life to sustain. And then, yeah, I find it hard to like even ask evolutionary style, like from an evolutionary perspective, whether maybe this is a very stupid remark, but I'll still make it, whether evolution acts on ecosystems rather than individuals. But yeah, so I think our understanding of evolution itself is very primitive, and these are very clean, more clean. So fluid mechanical system I find to be more clean to answer those or even pursue those questions. And so I think biological pump is the place to even look for the actually basic evolutionary mechanism in life forms.
One more question concerning the material properties of these particles that you see there. Sorry. Concerning the material properties of the particles that you see there, are they known? Second question, is there a temperature effect on these material properties?
So to the best of my knowledge, this is the first time someone has looked at the rheology or the elastic property of the mucus in the open ocean. So people have create artificial mucuses and isolate them in the lab. Also, one important thing to say here is our understanding of how biology or cell biology plays in is based on a very few things that we have been able to control in the lab. They lose their behavior once you bring them in the lab and let them stay there for long. That's why there is a value in actually going and looking at this in their native environment. And only then you can say something about the sequestration in the climate context. So even if you extract mucus from a lab culture, doesn't say much. It depends on pH. Elasticity has been shown to depend on the pH of the ocean. And I'm sure that it depends on a lot of chemical parameters that are there in the ocean. It's possible that different organisms create different kinds of mucus. So that could also, it depends on biodiversity as well, cellular biodiversity in the ocean as possible. But there are no clear answers to those questions.
Wonderful. Thanks again. Oh, sorry.
No, please.
Didn't see you.
You still have time.
It's okay. Well, it's a very simple question. Do you know exactly when the mucus forms, or does it change throughout its? Because you gather it from the bottom, right? So
I have observed or made these observations again in a different expedition. So if you look at a cell for a long time, so you start seeing that as they sink, they release. So you start seeing these beads, PIV beads, getting stuck on the surface, and you can see the halo being formed at the cellular scale. If you look at individual chain-like cells, is what we looked at, rhizocylene. So yeah, it forms from there, but at the same time, are we stressing the cells when we take them out? We try our best to do the observation immediately, But yeah, what leads to mucus formation? I do not know. Maybe someone in the biological community has figured it out.
Thanks.
Okay, no more questions? Thanks again.